Good afternoon. Howdy. Y'all are getting good at that. That's awesome. So just in case you missed some of the earlier sessions, I want to take the opportunity to repeat some of the thanks that we need to give out to the people that made this happen. Of course, Dr. Scowls, this is his brainchild and did the heavy lifting of getting these folks here. This doesn't happen without fiscal resources, and so I want to make sure that I thank my College of Veterinary Medicine and Biomedical Sciences, the college level and all 5 departments contributed. AgriLife Research contributed handsomely. The Hagler Foundation, I think Jim Junkins is here. Thank you so much for your generous support. And Faculty Affairs were the 4 big donors. I'm much appreciative of all that. And I'd be remiss if I didn't thank Dr. Ashley Seabury and April Orcutt from my Office of Research and Graduate Studies that took care of all the 8 gazillion other things that had to happen. So let's give them one more round of applause. All right, so I've been very excited about this session today because this is what you don't know what's going to happen. And I think this is going to be really exciting. I'm going to briefly introduce 5 panelists to come up, and then I'll welcome Dr. Kathryn Meurs back up to moderate this session. The title of the session is going to be Issues in Clinical Veterinary Genomics, and we have a very distinguished set of panelists. First up, we have Danika Bannasch. I called her Danika Patrick yesterday in a bad fit. Just as pretty. It is a professor of the Department of Population Health and Reproduction and associate director of the Center for Companion Animal Health and associate dean of the School of Veterinary Medicine at UC Davis. Her research is focused on the genetic basis of inherited diseases in animals, particularly dogs and horses, as models of human inherited disease. Danika? Secondly, we have Dr. Gus Cothran, who's a professor emeritus at our College of Veterinary Medicine and Biomedical Sciences with a focus on equine population genetics, conservation genetics, and genetic diversity of both domestic and wild horse breeds worldwide. Dr. Cothran? Third, we have Dr. Brian Davis. He's an assistant professor in my Department of Veterinary Pathobiology with a joint appointment in the Department of Small Animal Clinical Sciences. He's the director of the Veterinary Medical Biorepository in the Office of Veterinary Clinical Investigation at our VMBS. His research focuses on the field of comparative genomics, and he has been instrumental in addressing heritability diseases in dogs, cats, and other animals with the potential to translate these findings into treatments for similar human conditions. Brian? Fourth, we have Dr. Molly McCue. She's a professor and former associate dean of research in the College of Veterinary Medicine at the University of Minnesota. She has established herself as a leader in the field of veterinary genetics with a particular focus on metabolic disorders, performance traits, and inherited diseases in horses and dogs. Thank you, Molly. And fifth, we have Dr. Terje Raudsepp, professor in the Department of Veterinary Integrated Biosciences and the director of the Molecular Cytogenetics Laboratory at the College of Veterinary Medicine and Biomedical Sciences here at Texas A&M. She specializes in animal cytogenetics and genomics with a particular focus on equine and comparative genomics. Known for her expertise in chromosome biology, she's made significant contributions to understanding the structure, function, and evolution of animal genomes, particularly domestic and wild species of equids and camelids. Terje? And it's my pleasure to welcome back Dr. Kathryn Meurs.
All right, thank you, everyone. So this is really meant to be a group discussion of topical questions the audience might have. So I thought I'll try to throw one out and let's just roll with it a bit. But also, as we get going, if there are people in the audience that feel you've got a burning question on something you would really like to hear the panel weigh in on, please jump in and let's make this as interactive as it can be. So I think I would love to hear anyone on the panel just talk about what they think the biggest issue with moving clinical genetics forward to have increased use, increased application in the next decade will be. What's the biggest problem? And anybody can roll with that, and we can have, there may be everybody wants to answer it. I can see Molly warming up.
I was just saying I'm not an extrovert. I play one on TV, like when we're sitting back there. But, I think there's a couple of issues, and to me, they're pretty equal. I think one is making sure that we're validating the methods and we're validating the things that we find, not only for individual patients, but as we use them for multiple patients. The other thing I've thought about a lot, and I think we've talked about this probably 10 years ago now, is that we need in veterinary medicine, I believe, a community of practice around genetics and genetic counseling, right? You talked about it in your talk. There's so much misinformation. We're coming out with things that are, multiple genes. How is that interpreted? What about when we get to the polygenic traits, which is some of what we're doing in horses now? We don't have a community of practice. And part of that is obviously the science part that we're all a part of. I'm also a clinician and a clinician at heart. And so we need more people to bridge that gap. And I think if we had people, especially on the veterinary clinician side, right, that joined that community of practice, that would make a big difference.
So
Yes.
I definitely agree with Molly, of course. Work together on multiple things, we think, and I'm learning from her as we go. But I do want to say something that when Richard showed, he's showing numbers in human studies that exceed 200,000 individuals, 250,000 individuals. And in my lab, in dog, we're at 3,000 individuals. And that's across the entire realm of dogs that have been sequenced. And we're getting published data and we're trying to manage that data. I think we should look prospectively at veterinary schools predominantly to just gather sequencing data, just, maybe as part of a client service. I don't know how hard that is to sell. This is kind of out of my wheelhouse, but just having phenotypic data that's tied to whatever veterinary records are accessible so that we can, as we look forward at these polygenic traits, we're going to need to train models. We're going to need to look at very detailed phenotyping information. So, at least in the studies we have in my lab, we're not looking at great sample sizes, right? Low hundreds is gold, right? We're dealing with, low dozens, which is not a very powerful association. So I think we should really engage, as Molly said, with clinicians and try and just capture those client data. It's very inexpensive to sequence now, short read data, right? It's very inexpensive. So if we can convince clinicians to engage with the clients and just capture that genetic data, whether or not it's useful to us right now or not, we should capture it prospectively.
Well, rather than say I'll agree, because I'll do that a lot, I'll just say that when I first came here to A&M, I came into a veterinary school, whereas before when I had been in Kentucky, we were in kind of a pre-veterinary program. We weren't actually training veterinarians, we were training people who would go on and become veterinarians. But one of the things that disappointed me when I came here was that Jim Womack, and some others had talked to the administration within the vet school about more veterinary education for the vet students. And the basic answer we got was they have too much to do now. And we can't really incorporate. We had a genetics course that I and several others that have been here have taught for the undergraduates, but the people who were really aiming to be vets weren't getting any genetics except for what they had as undergraduates. And if you're going to start employing that practice, that knowledge to clinical situations, you really have to understand what it can tell you and what's available. Another thing that I think we need to have is simply develop more clinical tests. Now, I think all of us that are here on the panel and a lot of the people who have spoken or even just in the audience have worked on particular genetic traits that would have clinical value in a veterinary setting, but there's still an awful lot to be done. But probably someone like Brian would argue that let's get the sequences, and this kind of relates back to some of the things we heard today, is that once you have the sequences, then you can start looking for the genes. So we've got a lot of work to do in just building us up to the point where we can be effective in clinical treatment.
Danika?
It says on, but you push it away from the on. So I guess maybe in response to Gus a little bit, I do think it's important for veterinary schools to consider attempting to have a clinical geneticist on staff, because that person then can talk to the clinicians and maybe talk to other geneticists and really help get that knowledge back into the clinics, and to the students. We used to have a course. We don't have a course anymore. We have a new curriculum. We have 6 hours of genetics for our vet students. They absolutely are not experts in 6 hours. In 6 hours, they barely know what they don't know. And I would prefer that they not counsel with 6 hours of education that they don't remember. So I think that having a clinical geneticist at vet schools is a way to sort of get that information out and maybe educate the students. I mean, if they hear it in senior clinics while they're looking at, a litter of puppies where 2 of them are abnormal, that it's going to stick a little bit better than trying to lecture at them or do it in the curriculum.
This education piece, I think, is really, we have what's called a selective, so they can opt in, and I think it's limited to 20 people for a short course at the end of the semester. And so I think we really have not, as a profession, met that very much. There's not a lot of continuing education on it. I'm just emphasizing what a great point I think this education is, because our clients use these web-based tests all the time. And so in our selective, we talk to students about how they're likely to come in with the results of their wisdom test and want results from it. So it's an issue. It's a great point.
Well, education is indeed, yes, it's an important thing, and that's why we collaborate with clinicians, so they can tell to their students what to do, and we get many samples just through our clinical colleagues.
Yes.
Both in UC Davis, at Texas A&M, and other universities, even though we are not clinicians. But as I am a clinical cytogeneticist, so I'm a little bit different from these people, because they want to sequence whole genomes. We can give answers, whether there is a chromosome aberration or not, in, let's say, 5 days. You can't get the whole genome sequenced and analyzed in 5 days. So we have a little bit different old-fashioned approach, but so far there is no genomic approach which speed-wise, financially, or even in contents can beat the clinical cytogenetics. They can be combined together. Cytogenetics will be much richer with genomic data, but I don't see yet that cytogenetics needs to be completely replaced by genomic methods. For example, Illumina sequencing cannot detect balanced translocations. It can probably detect aneuploidies, but not balanced rearrangements, and of course structural rearrangements are beyond Illumina sequencing. And I see that we need, at least in United States, next generation of cytogeneticists, because we are the only lab in US, and I'm getting old, so.
Can you speak a little bit more about the cytogenetics aspect? I don't think, from a veterinary perspective, we haven't talked very much about that. We send our samples occasionally to you for that as well, because you're really, A&M's the only group that has that. And I don't think that is on the radar of many of us, many clinicians, about looking at those type of cytogenetic impacts. Can you share some of the things you found? Do you see those things more in cats, dogs, horses? What sort of species are you most seeing true defects?
Well, we see all domestic species and many wild species, from zoos, from wild animal parks. And the reasons usually are reproductive problems, occasionally congenital defects, but these are usually so severe that animals are euthanized before we get anything to do with them. But, well, thanks to Jim, I'm also not only in comparative genomics, but in comparative cytogenetics. And not only comparative cytogenetics, comparative clinical cytogenetics. For example, X monosomy. It was discovered in human, well, the first time the phenotype was described, I think, 1948. It was karyotype 1959. And only now we start seeing X monosomy patterns in animals because data has been accumulated. We have been around 24 years. And X monosomy is #1 condition in horses. All chromosome abnormalities, X monosomy is #1. It is #3 in human sex chromosome aberrations. And it is extremely rare in all other species. So why? And here comes in this work we have done with Bill on sex chromosomes. It is the pseudoautosomal region. Bill had a question about it in the morning. So this pseudoautosomal region is 1.8 megabases in size in horses, 2.7 in humans. So it's just a megabase larger in humans. And it is magnitudes larger in species we know where is the boundary. For most species, we even don't know where is the boundary. So we can't measure the part. And it is a clear correlation between the size of par and the number of viable X monosomic individuals. The smaller the pseudoautosomal region, the more viable X monosomy is. There is also an argument that maybe you see X monosomy more frequently with small par because the recombination, the size where X and Y chromosome can synapse and recombine is also smaller, so mistakes can happen more frequently. It may be an issue, but not only because in mouse, pseudoautosomal region is 700 kilobases, and X monosomic mouse are not only alive, they are fertile. All other X monosomic individuals in any species are sterile. So X monosomy, well-known condition in human Turner syndrome, it is so different in different species cytogenetically. So that's just a small, short answer to your question.
Very. No, I think that was very, very interesting. It was perfect.
I guess I wanted to say one more thing. I think all of this comes back to what, when I say community of practice, I envision, right? If we had people in vet schools, if we had a body of knowledge that we agreed upon was important, there would be people talking about cytogenetics and that we need that and what samples to send. There would be people who could interpret the genetic test and do the genetic counseling, because I think similar to everywhere else, I think we're down to 8 hours of genetics in the veterinary curriculum. And we have more and more discoveries all the time, and people don't know what to do with it.
Well, I can add to Richard Gibbs' story about somatic mosaicism. We recently had a horse with two different, the mosaic for 2 different translocations. So this you cannot detect by Illumina sequencing. You really need to do karyotyping. So, of course, we don't find all causes. Most animals we analyze are normal, but, well, like mutations, chromosome mutations are rare, that's why.
Or was that a symptomatic patient, the horse?
It was a potentially fertile mare. She had sometimes functionally active ovaries, but not always, not every season. She has had miscarriages, typical to balanced autosomal translocation, but we never seen balanced translocation in a mosaic form. She didn't have any normal cells. She had just one translocation in about 50% of cells and another translocation in others.
Fascinating. I'm going to check in a little. Anyone in the audience have? Yes, Jim. Lauren, sorry.
So as Gus alluded to, there's several individuals here at Texas A&M that have attempted over the years. I think probably Joe Templeton was the first one. Jim Womack was the second one. I tried, Banu Chowdhury tried, a number of us tried to get a genetics course integrated into the professional curriculum with little to no success. And the reason we were given was there wasn't room in the curriculum for another course. So I've thought about the alternatives to that, and I want to ask the panel what they would think or whether their institutes have considered this. And I say that recognizing that the representatives on the panel represent basically we don't have anybody from Cornell, but otherwise, those schools represented by the panelists are leaders in the field of veterinary genomics. So I would ask the panelists if their institutions have considered developing a residency program in clinical genetics versus a change to the professional curriculum. And if not, why not?
Well, so first of all, Penn, who's another leader, did have a residency program for a long time, and the residents in that program took care of the animals with inherited diseases. Most of those were mucopolysaccharide storage diseases in the colony, and then also any genetics cases that came in. I know that because I was going to go and do the residency, but Davis decided I would make my own residency at Davis. And I think we had one other resident afterwards. We struggled with bringing in breeders because we don't have small animal theriogenology. So if you don't do small animal repro, the breeders are not coming. And so it kind of backfired a bit in that it didn't go any further. But we did develop one, and I think that, it's something that had the potential to educate more people. I think that there's, it doesn't take, if you have a PhD in genetics and a DVM, you're a clinical geneticist. I don't know that you have to do advanced, residency training to be able to counsel. We have a decent number of people with that training and background, and they're situated different places and can provide that advice.
Yeah, Laura and I thought about this a lot, right? It's not something that we've done in Minnesota, but I think that more adoption in veterinary medicine comes sort of from students being taught, right? And how do we teach them? And to me, I think that is doing that sort of thing, right? Having some sort of specialty training or some body of knowledge we agree to is clinical genomics. I think maybe one of the things that's been missing is most residencies are ultimately towards getting board certification, right? So you can demonstrate that I have this set of knowledge. And so one thing that I've thought about, we've talked about, and Kate, we probably talked about this before, is do we need to have a specialty to attract more veterinarians, right? To look at this field, to understand, to start to infiltrate. And we need to give them a carrot, right? How do I say I have extra training? if you have a certification because you have a body of knowledge that maybe would make a difference.
I think the American Kennel Club created, I was thinking to Danika's point about the link to reproduction. So the AKC funds theriogenology residencies, and they've tried to fit in to a few of them a emphasis of genetics. So asking if you're one of their funded residents. We actually had a program with them for a while that, and I think most of the schools have a similar thing where we ran a hotline for genetic questions for the public. We would get maybe 1 or 2 a week. They weren't that frequent, but they were sort of interesting, and those would come in and go to the Therio resident. Challenges, AKC's funding them, so they want them really to focus on dogs, et cetera, et cetera. They're these things. But I think our specialties have tended to think, oh, that's a reproduction issue, and lumping them with Therio. Often the people that are choosing to go into theriogenology may not really have an interest in genetics.
They think it's a medicine issue, right?
Yeah.
Right? So the repo people, like, that's a medicine thing.
Yeah.
Those are internal
Yep.
medicine diseases for the most part.
Yep. It's very interesting.
We have just the opposite experience with Brian. We were in the committees of two residents who did their PhD in molecular genetics. And now they are working back in clinics, but they know a bit about genetics
Oh, that's
as well.
wonderful.
So it's both ways. Yeah.
Large animal theory.
And they are large animal,
Yeah.
both are equine terrier
Yep.
people.
Yes, please. Oh,
Thank you. So I have a question about the role of developing novel animal disease models for the sake of understanding their human counterpart as a driver for the general enterprise of genomics and genetics and animal science versus what I think you're all talking about is animal health, essentially, and through genetics. And I wonder about the relative role of each of those in driving the general enterprise.
Well, I'll say that certainly when I write an NIH grant, I definitely mention that the driver is to develop an animal model. It's interesting. I think over time it's improved, but we also fight a lot of sort of pushback from the human side that there's no point in doing this in large animal models, or dogs aren't good models. I mean, I can't tell you the reviews I've gotten over the years. And the one actually is, I think, the best example, since you brought up SLC2A9 earlier. So I identified the mutation that causes hyperuricosuriae in Dalmatians with an R21 from NIH.
and I should have linked that to you. I was
It's
just.
okay.
Sorry.
And I applied for an R01 to follow up on it, and I was told by the urology study section that SLC2A9 had absolutely nothing to do with the human condition. And we were talking about positive human GWAS, and if anyone wants to go and see a really amazing positive human GWAS, it's for serum uric acid levels, which is associated with SLC2A9 variants. So, you know, they're just, you keep getting this like pushback. Well, Dalmatians have nothing to do with humans. Okay. So, you know, we believe it, and we try and demonstrate it over and over and over again. And sometimes human geneticists believe us, and sometimes they don't. And so I think it's an uphill battle from a funding standpoint.
Yeah, and I would echo it. I would say that all of us up here, for the most part, have tried to leverage that, right? Have had funding from NIH or other places. I think we also recognize that while we're passionate about animal health, one of the ways to have funding, one of the ways to sort of validate the work that we're doing and being able to follow things forward is to show that they're a model of human disease. And certainly, probably all of us here have had NIH funding to do that sort of thing. But as Danika said, it's a hard road, and dog models are a lot easier than horse models, like we've done it. But, and there are a lot of really cool things about muscle physiology, exercise physiology. There's not anything like the horse to study those things, and they're important, but there is this sort of belief that their utility is minimal.
The other, can I add one other side of this too, I think, is that we are veterinary colleges, so most of, I think we're all started from the veterinary aspect of it. That makes it sometimes very difficult for us to think about, like for me, am I going to have a colony of Doberman pinschers, even if somebody will fund it? I want to make sure I can convince myself and our college stakeholders who come here because they love pets, why I'm keeping these dogs in a research environment. And so sometimes that is challenging. And especially again for our stakeholders, of why would you have a colony of research dogs like this? The other, but I've tried to argue, I think most of us have, is people are not living in a cinder block room, so wouldn't our studies be better anyway if we're following these diseases and they're out there breathing the air and drinking the water? The issue I got with NIH and funding is, the study section would like biopsies, endomyocardial biopsies at these time points, and sometimes it's hard to get those with people's pets. So that animal thing is sometimes, how do you walk that line comfortably for you and the stakeholders?
Now, Kate, I would keep your colony of research Dobermans. No biopsies would be happening.
In Molly's house, yes. Sorry.
I mean, I already have 8 of them, so we're on the way.
Yeah, so thanks for that depressing answer. The world changes quickly, and, the number of unsolved, human genetics conditions, which we've got the gene, but we really don't know anything about it, and the improvements in genetic engineering and the failure of the sort of traditional models has got to push things in your direction.
Right.
One would hope.
An example,
Thank you.
right at the same time, the very first loss of function mutation in glycogen synthase was identified in humans, we identified a gain of function in the horse, right? Still, that's never been shown. And it turned out it was an area of glycogen synthase, which is the most well-studied biochemical enzyme, 13 amino acids that are completely conserved. Horses have a base pair change, and it changes their muscle glycogen synthase 1 physiology dramatically. And we have applied for funding to try to follow that up and say, look at exercise, why, when they make so much glycogen and break it down, do they have the skeletal muscle energy deficit? Why do they get rhabdomyolysis? Is a really novel model, and trying to convince NIH that it was useful has been extremely difficult. And it's hard when I can write 2 NIH grants and not get funded, but I can get just as much money by getting a bunch of USDA grants and foundation grants, right? At some point in time, you say, I have to fund my lab and pay for my students, but I do think there's opportunity. We have to sort of be better at selling it, maybe.
Yeah, nothing positive to say about NIH review grants.
Maybe we
Especially right now.
Maybe we could talk about how we fund these things, though, because I do think that is a real issue. And Molly, you're saying the money's similar, but so USDA will do horses, but for dogs and cats, and this, I'm not complaining, I love what I do. So, but most of, for dogs and cats funding, unless you're able to use it as a model for NIH, it's generally maybe $80,000 for a 1 or 2 year grant through the AKC or Morris Animal Foundation.
Yeah.
We'll sometimes do a bit more, 100 or 200,000. But again, they're wonderful that they do that, but that does limit how many whole genome sequences you're going to do, or some of the proteinomic, you have to make these decisions. Again, not a complaint, but I'd love to hear some of the others talk about how they fund these things.
I come from a natural population, wildlife background, and not a domestic animal one. But when I started working with horses, was actually, the work I was involved with initially was actually NIH-funded study of standard bred horses as a model for human reproduction and genetics. Wasn't a very good one, but it did fund a nice study. But then when I came to Kentucky and we started looking at the horse as an agricultural animal, at that point, the USDA was not funding horse research. It was a companion animal. If you went for a companion animal, no, it's an agricultural animal. And so you had to struggle, same kind of place that you just talked about, the horse organizations, Morris Foundation. The grants were very small and not for very long usually, so.
To put that in perspective, I have a little over a dozen people in my lab. I was putting together my annual report of accomplishments for the college. I wrote 23 grants in 2024. You know, and I think right now I have like 18 funded grants to pay for all of that. So I think just as hard about those grants and what I want to do and putting them together. There's not as much extra paperwork as there is for an NIH grant or a USDA grant, but like it is hard and it is a lot of work, and you're kind of cobbling big projects together by peeling out this small piece that Morris might buy and this piece that AQHA might buy, or maybe NSF will be interested in this from the genome biology, or this piece is a model. And so you put together a program, but it's tough because the number of grants that you have to write to do that is a lot of late nights and weekends.
I mean, I don't know how much more I can add, but yeah, so far my successful funding has been from foundations, right? You know, it's animal specific, very little application to humans. We hope to change that maybe in 3 years, and I don't know how. But you know, Terry and I have had successful funding through USDA for horse work, and you know, I'm a co-PI on a grant with Molly. So it really we have success when we work together as a collaborative unit, right? So, but it's dismal. I will say it is. It's harder, I guess. Still enthusiastic about it. I'm not jaded or anything, but.
I think we do that better in the horse genomics community than maybe the dogs genomic community too. Like
I think
a lot of the things we've accomplished in horses have been the whole community together.
This was noticed already at the time of, well, comparative cytogenetics and zoofish. Dog people were fighting with each other. Horse people were collaborating. It has a long historic.
So I sort of jumped ship from
dog
horse
people
to
very
dog only when the tools got better in dogs, but then the horse people are nicer, it's true. So then I kind of go back because.
But Richard, in answer
Yes.
to your this proposition, animal models, I think cat and dog has been proposed a good model for human asthma, like equine recurrent airway obstruction and metabolic disorder in horses. Molly has a long experience with that, like a model for human metabolic disorder and diabetes too. But I don't think we really get NIH funding on that.
I successfully argued that dogs were a good craniofacial model for human disorders and cleft lip and cleft palate. So I got that funded. It's convenient that mice eat their cleft offspring, so you can't study them because they're gone, and dogs don't. So that worked out well. And I did use the US Postal Service to have people around the country mail me dead puppies, which is probably illegal, but we won't talk about that. That worked really well. We found lots of really interesting variants that worked. But.
Is there a question over here?
Yeah. I want to perhaps not close this session out, but add a missing link. We need an aerospace engineer to ask a genomics question. At an event here about a decade ago, we had our guest of honor, not the president, Jim Womack, hosting a conference, and he had Huda Zogby from the Baylor College of Medicine, and she's an expert in a specific class of inheritable neurological disorders, and in fact, she is, I think, the world expert on Rett syndrome, and in a strange set of coincidences, my best friend Clifford Fry's daughter, Ashley Fry, was the first Rett case diagnosed 45 years ago when Huda was just starting her career, and her obsession with Ashley led her down a certain path. I certainly am not an expert on Huda's research, but I recall her presentation and conversations with her that Clifford Fry's wife, Judy, has the Rett mutation in her genome. She had one very healthy daughter without Rett syndrome, and she also has a genetic mutation. And to have Ashley, which has Rett syndrome, which is very debilitating. And a large fraction of Huda's research, again, as this amateur aerospace engineer understands it, has been on what controls gene expression. And I think she discovered what controls the expression of the Rett mutation. And it's a certain protein, I can't begin to describe it, but she has induced and in some sense cured in mouse models Rett by controlling the presence of this protein at the judicious times. My long-winded question is this: gene expression or not has got to be an issue on both human genomics and all of the genomics associated with veterinary medicine. It would seem to me, if I was at NIH, I would entertain a proposal. Get after it, young man. So I think seems to me as an outsider, there are huge interfaces to be explored, and that's just my thought.
Absolutely. One of the other things that Terry had mentioned about the horse is that they're long-lived, 30 and 40 years old. We don't have another animal model readily studiable that's like that. We look at a disease that has the same signatures of neuronal body degeneration and alpha-synuclein. It causes a very different phenotype because of where it is in the brain, but we argue it's a Parkinson's model, and when they live, it's a disease of aging in the horse as well. And when they can live 20, 30, 40 years, then you can really look at something that's happening over time and slowly.
Yeah, I mean, so we, both in the horse and the dog, we have, I think, just recently developed in the dog and the horse a little bit earlier, but annotations and expression data from multiple horses, multiple tissues, and in dogs, I mean, I think it was like something around 350 different tissues from a series of dogs. So we have that available to us as a tool. We still suffer from some annotation issues in our genomes, unfortunately, and particularly in, I think, less of a problem in dogs than in horses than it is in dogs, but I don't know if you notice, dogs look really different from each other, and just picking one random dog to be the reference genome has definitely caused some issues that we still try and recover from. So. And a boxer, which if any of the veterinarians know, boxer was not the healthy choice. They're cancer factories.
And cardiac.
Brian's going to take care of, and cardiac, sorry. And Brian's going to take care of all that now, though, and tell us the solution.
The solution. So, if we go back to studies that have occurred over the past 15 or so years, we find regions, we don't find really necessarily mutations, right? I think what Richard showed was, well, we do. I know, Danika, I know.
We sometimes find mutations.
We sometimes find mutations. That's very true. Specifically for Mendelian traits, but especially when we get into polygenic traits, it's going to be gene expression. It's going to be regulatory regions. It's going to be structure. It's going to be genome structure, which Danika brought up. Most people are using one reference genome. We need to look a little bit past that, because particularly in at least what we've seen in dogs and also in horses, structure varies, genome structure varies drastically between single individuals. I mean, we see, right now we're building multiple reference genomes, and my lab over there is working on it. Sam over there is doing it in horse with Molly's group, and Sarah, I don't think is here, she's doing it in dog. We're building multiple reference genomes. So we're actually going to try and look at the structure of the genome in the context of regulation, right? Or in the context of gene loss and gain, right? So we think there's going to be a lot of information there that's going to show up over time, but we have to build the resources to do it, and where's the money to do that? It's a very expensive proposition, but working in groups is how we end up doing it. But yeah, I think a lot of the answers, especially the polygenic traits, 100% is regulation of gene expression. It's not a protein coding change. Yeah. I'm sure Leif has a comment.
So I don't work in clinical veterinary genomics, but I should just share my gut feeling. And so my feeling is that, so we're talking about the future of clinical veterinary genomics, maybe particularly in companion animals. So I don't think the solution is to do more genomes. I think, in fact, that what the best you could do to improve this is to work with the breeding organization to implement sound breeding programs. You could develop a really nice SNP chip panel that you could use to do genomic selection. We know that's not rocket science, but we know that it will work. You will be able to breed out defect variants, although you don't know exactly which variant, if they are a small effect variants. You heard from Richard about the enormous investments which have been made in human genetics to try to nail down this causal relationship between sequence variation and phenotype. And the question is, do we really have those resources? The problem we have is that in our domestic animals, they are split into many breeds, and each breed is quite small, and you have. But on the other hand, you could say the advantage with small breeds, you have a lot of LD, so it should be easy to establish those haplotypes that is predisposing to disease.
I would comment on that because I agree with you completely. What we're doing in horses right now and all of our polygenic traits and dog behavior and what we're trying to do in the Doberman now, we're getting thousands of Dobermans, is develop predictive assays, right? We don't need to know all the genes to make a difference in breeding, right? Knowing the genes, I think, is still interesting because when you're targeting therapies and such, but I think every grant we've written for a horse disease lately has been, in this aim, we're gonna create a polygenic risk model, understand selection, how to breed away from it. This aim, we're gonna look at the biology. And we need to do both, because if we wait for all the variants, it's gonna take us forever.
I agree. I mean, one should be opportunistic. There is an opportunity to learn some really interesting stuff, and then you should take that opportunity. But if you think about how you improve health, I think you need to have sound breeding programs.
So I'll just talk about dogs since you talked about horses. The problem isn't, and I'll give an example from the Nova Scotia duck-tolling retriever where I've identified 8 different mutations for Mendelian traits, and while I was sitting here, I got a 9th one that I'm gonna sequence when I get back. So they're, except for one, they're recessive, they have the tests, they can select, and there's still diseases in the breed that are not tractable, they're not Mendelian, there's autoimmune diseases that are not simple. They started from a population of 3 dogs and 6 full siblings, and there's now 50,000 Nova Scotia duck-tolling retrievers. Most of them are in Sweden, by the way, because the Swedes love this breed. So it doesn't matter what you, there's more mutations to be discovered, and I don't think that you can use breeding values when you have so little genetic diversity. So I would say that the answer is opening up the stud books and not having closed populations. And then you can look at another breed, the Danish-Swedish farm dog, which is from Sweden and has almost no inbreeding, and they're perfectly healthy, wonderful animals because they're not inbred. So the answer in dogs is, it's absolutely obvious. And no, you don't need to find any of the variants. You just need to start breeding them in a diverse way, and all of the bad stuff will go away. The cancer and everything else that isn't Mendelian, that we haven't solved yet. right? And if that wasn't true, then we wouldn't have these incredible breed predispositions like boxers and golden retrievers, sorry to the golden in the room, and golden retrievers to cancer. And, you know, that's all breed specific because there is an inherited component because they are 25% homozygous.
And maybe, Danika, and I wanted to go back to your Dalmatian story because I think one of the things my understanding is that we can recommend more outbreeding, but they're often the breed organizations that have on a national level or an international level that have really strict guidelines about what is allowable. And so I think to move away from them, they essentially have to stop using them. But maybe you could talk about your experience with the Dalmatian, if you would, and maybe I'm misremembering some of that too.
Oh, the pain and suffering?
Well, your suggestions on moving it forward. And
So the breed was homozygous for this mutation that predisposed them to urinary bladder stones. And an amazing mouse geneticist, Bob Scheibel, who worked on the same mouse mutant that I worked on for my PhD, which is super cool, decided that he could fix Dalmatians by crossing to a pointer and then back crossing to Dalmatians. So he introgressed the wild type allele. And at some point he got them accepted for registration, and then he went to a dog show and he beat some purebred Dalmatians, and the registration was then rescinded, and he wasn't allowed to register the dogs for about 30 years. And then I came along in the middle of vet school and said, hey, I'm gonna find the mutation for this. And there was a group of Dalmatian breeders that said, wow, if you find this mutation, we're all in. We're gonna participate and help. And then ensued a 10-year battle with the parent breed club who did not want to register the dogs. And eventually, and I wish Elaine was here, eventually AKC convened a panel of genetics experts to look at all of the data, and the genetics experts, which included Elaine Ostrander, said, yeah, register them now. That's as ridiculous. And so they are now registered. They're registered with a scarlet letter so that all the purists can avoid breeding to them, and all of the people who want healthy Dalmatians can know which ones to breed to. And you can have Dalmatians without hyperuricuria. But it was literally a 45-year battle over a simple Mendelian trait, let alone something as complex as cancer.
which easily, if you'd been able to outbreed, would have taken care of it, it sounds
Well,
like.
it was, I mean, the backhouse dogs were successful.
Yeah.
I
So
would
it was
say-
ugly. And that was because of the breeders, not because of the American Kennel Club.
Right.
Okay.
I would say, I was talking to Kate about this over the break. So I breed Dobermans, and I happen to be totally in love with one of the most unhealthy dog breeds there are. So Kate talked about their cardiac disease. It kills 58% of them. It's like 4 times more likely than their second leading cause of death. And it's a big problem. And we have liver disease that 30% of them have. It kills a lot of female dogs, and several autoimmune things. What's interesting is that we're finally, yeah, and I won't even talk about, we could keep going on. The wobblers and autoimmune. What's interesting is that there's some recognition now in the breed, right? And when I am personally involved with the National Breed Club, as we're talking about, we need to look at this. I just got some funding from the Breed Club to look at a population viability analysis in the breed, right? Can we show, like, I won't be surprised if it would qualify for being in the extinction vortex, right? If we borrow from wildlife ecology. But I think the reason they've given us funding to do that is because they recognize that the breed's not going to exist. And we have a large project we're doing in the breeds called the Disappearing Doberman Project. And our tagline is like, if we don't do something, we won't have them. And it's a little bit different. And they're thinking about like introducing this idea of, well, if we're in an extinction vortex, then we have to crossbreed. And the conversation is about which breed do we crossbreed with, right? It's not everyone, I'm not saying it's going to be adopted, but the Dobermans are in such bad shape that at least there's some open-mindedness about that. So bladder stones, I guess people don't care if their dogs have bladder stones. If they drop dead, they care a little bit more, maybe.
Okay, hold on a minute. You drop dead because your bladder bursts if you're a male Dalmatian.
Well, yeah, that's true. But is it killing 60% of them?
My sense is that in time, people, I mean, in the end, with most of these, I feel like we eventually get there, that people start using the tests and they're more comfortable with it, but it takes years, is my sense.
Okay, so you said you had this pipe dream that people would use the tests.
Yes.
I've developed 20 tests for Mendelian traits.
Yeah.
No one, not a single person, equine or canine, has gotten rid
Yes.
of the disease. They manage the disease,
Yes.
but they haven't gotten rid of the disease. And if it's dominant and therefore unacceptable to them, they say it's not real, or it's not a good test, or
Yeah.
because they can't actually stomach the results that if you want dachshunds that don't herniate, you can't have dachshunds.
Yeah.
Yeah, I would say like the Quarter Horse Association went down the road, right, of we're not going to register things with Mendelian traits. Well, the problem is we're going to keep finding more. And I think we talked about this before. You get rid of everyone that has as a carrier or has one of these diseases, you have a tiny genetic pool too, right? There's some balance between that, breed away from it, but do it in a slow and responsible way, or just crossbreed.
Yeah, I work with breed organizations that have the same thing. It can be done if the breeders and the breed organizations are willing to cooperate. I mean, one way to do it, if you have a single Mendelian trait, you don't have to not breed that animal. You just have to not breed it to another carrier, or if the offspring is a carrier, that animal's not allowed to breed. And, you know, you can do that, and then you can also do some other things where if you do other genetic testing, you can keep the gene pool variable enough to not start showing inbreeding problems, although nobody knows exactly what level that's going to be for any particular given breed. But the problem is getting the cooperation. And I've worked with breed organizations that are really foresighted. They have the best interest of the breed in mind, and they do a good job for a while, but then the leadership changes. And we've seen how that works.
I'm going to just check in the audience to see, yes, question in back, please.
Hello? Okay. So, there's been a lot of talk, about, we've developed tests, we can have a plan, we can have crossbreeding, we can develop ways to improve breeding standards to kind of remove those pathologies we're observing, right? And you're working with, breeders and breeder organizations. But, they have an incentive, it takes time, money, and effort to do all of that, right? And so in their mind, they're calculating that cost-benefit analysis, how much time, how much money, how much effort, how much messaging to, the wider public about, hey, these are the changes we need and they're necessary. And so that takes time, from scientists and from them to the public and them even internally. So that is, I think, in my opinion, a much more difficult proposition to develop a plan around, because you're working with those prevailing winds. So shouldn't we speak more to that than not necessarily the science? The science is important, and that will chug along because we're passionate and we will do that work. But the hard work is actually changing that cost-benefit analysis mindset.
I'll jump in. I agree absolutely, and I think that I think it's really important. It's just, as we know with any cultural, it's one person at a time, and it's one breed organization, it's one Saturday morning, derby or something, and it's really important, but because even what we have today in 2025, people could be using more. to your point, I had a call several years ago. We had a cat test for heart disease, and it was $25. And the woman had sent in some DNA, or she thought she sent in a swab, and there wasn't any DNA on it. And the lab had called her and said, in those cases, you do have to pay another $25 because we spend a lot of time checking. And she called me back and she said, I've still not made any money off of these cats. And so how can you expect to keep charging this? And I thought, that really helped me understand. I mean, it is for some of them. Some it's a hobby and they're just doing it. But for many of them, it's a hobby that they love, but they have to find that money somewhere. It's their hobby, but they have to find money for it, and it's expensive. And so if they're seeing some of these factors, they've never gotten rid of that disease, test cost has gone up, who knows what it means, they just pull away. And we could do a lot more if we can bring them along with us, but that is slow and it takes one person at a time. And I think, now I'm kind of running with a soapbox here, but things like social media have made it even harder. They'll say, I was on Facebook and my club blackballed me because I said this or that. So I think it's a very important, very valid point. Finding the time and the effective and efficient way to do that feels that it needs to be very human, sort of showing up connections, and that feels, it's slow, I think.
I think that's where we need veterinarians, right? That this is their specialty. I mean, we do this, Kate does this, Danika, I think we all do this. We talk to people as much as we can, but we're also trying to do the research and lots of other things, and it's slow, but we need sort of some bigger, I think, way to communicate, a bigger community that's doing that communicating, because every time you find something, you spend hours and hours and hours talking about it, and you can only do that so much.
So kind of to follow up, so maybe the next challenge, for us as a whole, as scientists, or at least people who are technical, it's not maybe so much the science, like that isn't the next great challenge. The next great challenge, in my opinion, has been, and increasingly looks like will always be, being able to communicate that. A lot of our research is, behind paywalls that normal people who aren't in this space will never have access to. And so then we cede that entire communication space to the world, essentially. In no other time in human history have we had the knowledge and the capability to learn something, right? But, to give that freedom, you can't curate it, but you have to speak louder and you have to speak more. And so maybe the challenge isn't creating more scientists per se, but creating folks who can speak to it.
I'll just say that, it's a very different environment in Europe. So right now, there's a really strong push in Europe to improve the health of purebred dogs by allowing outcrossing. Finland is the bastion of perfection in how dog breeding should be handled. It's all you, Terje, because they've opened up the stud book and for many, many breeds and said, go ahead and crossbreed and the stud book will be open and you can bring them back in. And that's to deal with breeds that are homozygous for multiple deleterious mutations. So that's happening all over Europe, and we're just a lot slower about things like that here. So, maybe that'll take hold.
Danika, I know why Finland.
Why?
Because Finland
Happiest
is
place
the
in the world?
leader also in human mutations because of inbreeding. Of course. Lena Pelto's name was mentioned here, that she was leading human genetics worldwide because they had so unusual, unique cases in Finland. So they know what inbreeding can do, and they are applying this to dogs. Good.
Also, a little shout out to Sweden, because the Swedish dog breeding is controlled by the government. You have to sort of apply to be a dog breeder, and you have to follow certain rules. And one of the rules is that you can't breed an animal that is destined to produce an inherited problem. And so the response to one of the dominant mutations that I've discovered is that they don't test, Because if you don't know that it has the dominant mutation that causes a problem, then you're not doing something illegal. Which is another interesting approach.
If you don't look, you won't find. Other audience questions? That was an excellent point. Thank you.
I was saying earlier that I would bring my 10-month-old Doberman puppy, but she'd be sitting in my lap, and so I'd be talking around her the whole time, but yeah.
Oh, there it was.
Every time I'm on a Zoom with Stephanie, she's mad that she can't see my dogs. And then I brought them to the hotel, just for you.
Please, go ahead.
Yeah, may I say that in your presentation and all the discussion, it's a remarkable similarity to what's happened in human clinical genetics. And all the issues of false positives and incidental findings and insurance payments, all the same. And actually, if each of your excellent panelists happen to be representatives of human healthcare payer systems, you probably would have said the same things about deployment in the human health. So, and to the excellent point just made about it's really not the science, you got to change the mentality. In the human area, I would say the single things that have changed the mentality are, one, the doctor, the physician asking for it, and two, the little bit of value that you can actually get from the genetic test. If the genetic test doesn't give you anything, very hard. If it gives you something, whether it's whatever that something is, then, so those two things are key. Anyway, just making a comment.
And that goes back to this education perspective that the panel kind of started with, and Molly just brought up again. The more we can bring more veterinarians along, the more we'll have that army to be out there selling this a little bit. It's great.
Yeah, we don't have a profession of genetic counseling in veterinary medicine, right? Those people who are trained in what they do is translate from the science to the clinic. We don't have it.
Dr. Adams.
Question I have is programs like All of Us, which I'm a part of, have been from the beginning. How effective have those been in identifying and understanding genetic disease in humans? Now,
That
I'll
question is
follow
not
that
for
while you're
us.
hesitating.
That is for you.
Okay, and I see Dr. Gibbs there, yes. I hesitate to ask that because why haven't we in the veterinary profession aimed higher, a moonshot, a Manhattan shot, at looking at this at a higher level where we collect a lot of data, the metadata, all the data that's associated with the case, including the DNA from that animal and everything that's involved in that case, and studied that across species. Okay, limit it to a few species and really dwell on that. There was an application in Germany across 7 different hospitals. They used something called swarm learning on 8 different diseases and 177,000 cases in humans. And from that, they identified many of the major risk factors and genetic factors included. So I wonder, in animal health, could we aim a little higher than here? All of the individual breeds, I've worked with those. I'm a pathologist, so I know a little bit about working with breeds and infectious diseases, but it just seems like we don't have a unified theory on how we want to attack the problem that's really captures the imagination of the public to invest, either through a government organization or through their own will. So how effective has Biobank, 23andMe, not 23andMe, All of Us, has that been useful? Dr. Gibbs, is it useful or not?
It's coming. Mike's getting his work out.
Well, I think that the realistic answer is no, not yet. But the aspirational goals are still valid. And the value of the collection and what we're accruing will make people like me who say the genetic risk is not explained, it'll be explained. Well, this is the vehicle for real explanation. And I think your point, I don't want to characterise you, but you all seem a little beaten down by the reality by these pragmatic issues. And yeah, it would, if you completely step aside naively and you say, what are the opportunities here? They are enormous, especially with the advances in engineering and especially with the other systems not reaching their full potential. So I think the right recipe can be composed to capture the imagination and garner the support and garner the funding. And if there's any lesson to get from all of us, it's like if you don't shoot for the moon, then you won't get to anywhere towards where you want to get to.
I'd say that at A&M we've had some success with this, with like the dog aging project, right? So it's pretty massive effort here. The tangible outputs we have really just kind of starting to trickle out now, but it's an excellent point, right? And just being able to collect the genomic data is for me as a comparative genomicist, evolutionary biologist, we need that, and we need to be able to tie that in much larger data sets. Like, Molly, you're thousands of Dobermans, right? It's going to be the biggest Doberman study ever by a long shot, right? So we have to start, and we have to work together to do it.
And I would say, Danika can probably speak to this too. Certainly we have tried to do those things at our institutions. It's hard to keep them perpetually funded because we don't have a lot of money in veterinary medicine, and the dean says, what do you have out of it? It's been 2 years. I think at Minnesota, we tried to do a couple of things. When I was associate dean, it was one of our big pushes. We developed a program where we're starting to learn from all our medical records with natural language processing, making our record system more mineable and meaningful, and then along with that, biobanking. But our small animal clinic alone has 45,000 patients come through a year. And so if you think about what it takes, just in tubes and freezers and infrastructure to save that, it's quite significant. And there's certainly conversations like through the COHA Biobank Committee and other places about how do we collaborate together as schools. So we want to do it, we want to think about it. In my lab personally, I became a faculty member in 2008. I think we have 21,000 horse DNA samples, which are horses that have deep phenotypes for different diseases. And the way that we found to be successful, which is what we're doing in the Doberman, is that we reach out to the owners and then ask them to provide the information in the medical records. Veterinarians are too busy. Academic institutions don't have the money. But we launched our Doberman study a year and a half ago, and we have 1,400 dogs enrolled, and they answered a 415 questionnaire, sent in DNA. We got all the medical records, all the Holter monitoring, et cetera, and we're asking for that every year. It takes dedication from those people, but that seems to be the way that we've been successful in doing it.
There's definitely different resources out there too. So Morris started the Golden Retriever Lifetime Study, which is 3,000 Golden Retrievers that are sampled and questioned yearly for all sorts of things, and that cohort is available to any researcher. They can apply to get different parts and pieces of it as appropriate. Those dogs are all on array, and the subset of them are being whole genome sequenced, and I think what they're trying to do is convince researcher X to whole genome sequence this chunk and researcher Y to whole genome sequence another chunk, and the dog aging project. So there's definitely some large scale ones, and then there's a group of veterinary schools that are part of this COHA Alliance that have a translational component, usually a med school on campus, co-located, and there's a biobank consortium through that, so that the individual vet schools who have biobanks can sort of be co-listed. At my institution, I started putting away samples a long time ago. I'm not going to say what year I started. And we have one of the oldest electronic medical records of any vet school. We started in 1988 with an electronic medical record. The problem is that we don't have SNOMED terms for diagnostics, and so it's a never-ending process. But we are working on that, and we have an investment
Davis,
from our dean in that.
Yeah, and like the SNOMED coding and stuff, we're working with Davis. There is a consortium on that side too.
We, I think a number of other vet schools do this. So we have a veterinary genetics laboratory that does genetic testing. I think when people were here yesterday, you heard about Stormont, who sort of started the VGL, and I don't even know how many millions of samples sit there that potentially could be used for research. They are swabs, so they're not like the best samples in the world, but so I mean that happens at other vet schools as well. So I mean, I think we do have samples, we do have medical information associated with a lot of them, and a lot of us use those for research projects. So I mean, I didn't mean to sound gloomy. I love what I do, and I think it's super exciting. And you know, I just wish the breeders would respond a little better.
Checking in with the audience. Oh,
What's holding it back from becoming a national effort?
Well, it sort of is, in that we know which institutions have biorepositories. So we don't have a representative from, is anyone from Cornell here?
I was going to say,
So Cornell
have
has a really well-funded biorepository. We have one at Davis. There's definitely some, you know, at multiple vet schools.
And ours is nascent, right?
and
We've
yours
had
is just starting.
multiple fits and starts, but.
Had multiple efforts to get them going.
I think I was recruited here once to try and
Unsuccessful.
get it going.
Yeah, when I first came, that was one of the things I was interested in
Yes,
doing, and we kind of reached out. Scott Dendo tried very hard when he came.
That's right,
Initially
Scott.
came here. And Brian has picked up the baton. I think the point there is that in the race, you can't drop the baton and then pick it up without losing a lot of your progress. So
And you said you were a pathologist, so I have to blame you, because putting everything into formalin does nothing for what we want to do.
Don't throw away pathology samples after 10 years.
At what point do you start to have conversations with breeders about breed deconstruction?
In the Doberman, I think it's now, honestly, and that's part of why we're collecting some of the data, right?
I think it also depends a lot on the breed. So the Dobermans themselves had broken out into this, they kind of have two groups, this united group that does more for athleticism and things. Many of the groups, it's very, very important to them that they're part of AKC and they participate in conformation, and they're each, that I think is a little bit of the challenge. It's not I don't want to sound downtrodden. Each one is an individual community with their own culture, and we've done studies with Dobermans and boxer people that have had amazing participation. I tried to do something with Rottweilers that was free, it was funded, and I just couldn't, they just didn't show up. But so my point is not one group's bad and the other isn't, but they are little microcosms of our cultures. And so I think that question is probably so dependent on dog, cat, what breed, what their goals are to do with their animals. That's my opinion.
So there is a conversation about deconstructing breeds, and it's happening. So you might all know that oodles are very, very popular, and the reason they're popular is because people have listened and have heard that dog breeds are unhealthy, and so crossbred dogs may be healthier. So that has swayed people to want to get those dogs. There's a group called the Functional Dog Breeding Collaborative that is trying to help people who want to breed outside of sort of a AKC breed, that want to breed just healthy pets for people, and trying to get them resources and knowledge, because there are a lot of resources within breed clubs that help people with their breeding practice, with selling puppies, with whatever, in addition to the competition. But so, I mean, it is happening. Like I said, it's, I think, happening more in Europe than it is here, but it's still happening. And I'm a wimp, so I switched breeds to a breed that has genetic diversity, so I could still play with the AKC sports that I like. The Danish-Swedish farm dog. I feel like I should have. It's a real breed. It's just admitted to the AKC with no inbreeding.
It's because it's more like a warm blood breed than most
doesn't
purebred
matter.
dog breeds.
The point is that the breed organizations and the people who breed them have to initiate those activities, and most don't. Now, I was just contacted within the last week by a very rare small group of horse breeders that have maybe 2 stallion lines left in their breed. They actually have better diversity than they think, but they're talking about who do we outcross to? And, it happens, but it's not common. And a part of that is that it's the specific, possibly single attribute of that breed that attracts people to it. And if you start deconstructing the breed, you start deconstructing the attributes as well. So it's a case of where we are willing, as academics and people who think about those kinds of things, are willing to work with those organizations, but they have to find us and tell us. And I have worked for a long time with the Livestock Conservancy and I've worked with rare breeds of various kinds, mostly horses, but some others. And they have those kind of issues in mind, but still the breeders have to initiate it.
I have to ask this question. When I was a resident here, I saw a patient that was like a Labrador and it was hairless and it was called a blue Lacy. And now that I'm in Texas, is that a real breed? Anybody? Yes, see, there. So you find these things, they're small and unique. And anyway, sorry for the digression. Additional questions from the audience? You go, Penny.
So in the spirit of throwing out a half-baked idea because, it just seems appropriate, we've been talking about, at the beginning we started off in the spirit of things, Stephanie, that came out of random conversations is where I was going. But
I threw out the blue lacy,
just
so
to
you're
clarify
okay.
with the blue lacies and all that. But we started out talking about education of veterinarians or what other models for education. And then we've been talking about some of the difficulties in funding to do the kinds of projects we'd really like to do in specific animal species for the sake of those animals, not necessarily because they're a model or all of that. And my personal bias is that one thing we've failed to do is really still get to function of the biomedical function. We can make lots of sequence and save samples, and we have to remember to do a good job of collecting phenotypes. But are we approaching a point that, and this is where the half-baked part comes from, or design your own dog. No, are we approaching a point where there are a number of veterinary schools that could maybe get together in a consortium-like approach and we find a way to collectively, I don't know, in this environment right now it's a little bit tough, but secure some source of funding where we could really start to do these things where it wasn't just a one-off with a breed association or animal foundation. So really, my comment is more to sort of plant some seeds of how could we work together and build this next thing that we'd like to see. And then in 20 years, we could say, hey, it started at this meeting.
I mean, that's one of the things I would love to see us do. I think there's probably a handful of schools that have genetics programs and also clinical people who are in the genetics program. And I think that that's what we have to do. And I think working together, thinking about how do we collect samples or data in a bigger way, but I also think how do we educate in a bigger way? And to me, that education, that community of practice piece almost has to come first because we have to get buy-in from those people who aren't looking at it from a geneticist lens all the time. But I mean, I think we're in the position to do it and that's what we should do. Where the money comes from, that's another story, but I think that's what we need to do. And I think the time is now to do that kind of thing.
Molly, if I can jump onto that, I like what you said, and Gus mentioned we're academics, and the immunologists, it took global pandemic for us to regroup and say, we have to do a better job of education at every level. And for us to go out, and vaccines didn't work, but vaccinology works, and there was a big difference, right? So it seems like there's people that want to use the right education, and maybe it's the breeders won't do it if their clients won't do it. And so, the immunologists, we decided we have to go to high school teachers, and we have to learn social media, and we have to do things just differently. Sounds like you guys already have traction in the right directions to maybe do these things.
Yeah, I think so. I think that concept of even high school, that's very interesting, sort of going even farther down. It's a great point.
I mean, and on that level, right, NSF is a place to look for money on the education front, right, to make that reach further back in training.
What? Okay.
So Dr. Meurs
Yeah.
presented in her presentation, she talked about the OMIA, which is a compendium of Mendelian traits in lots of different animals, including cats and dogs and farm animals and so on. Doesn't include QTLs and things like that. And it's largely a list of reports, and it's a compendium of that information. I don't think there's a clearinghouse that discusses whether a trait is the quality of the data for that trait, whether the variant has been proven to be causative or not, or whether it's in linkage disequilibrium. You're talking about veterinarians or people advising individuals about veterinary genetics. What is the source of information that they could go to? And again, information is developing rapidly every year. How do you keep up with it?
It's like I paid you to ask that question.
Each of us in this room has
specialty in clinical genomics.
Each of us in this room has special knowledge about some particular aspect, perhaps not everything. A cytogenetics question comes up. I know a little cytogenetics, but I know enough to refer everyone to Terje. Yes.
So we all refer to Terje.
So
ICE, it's a perfect segue back to where we started. So ISAG has a committee now that has recently been formed to provide evaluation of variants following sort of the human standards of declaring pathogenicity or benign function for the variants. And I want to say there's about 30 people on the committee. Kate?
Yeah.
30. And we are covering all species, working through OMIA, and actually going to have sort of a declarative information about all variants that are in OMIA. And I think ISAG is a really great organization to think about tackling that. Do you want to add any more about it?
No, well, the only much more superficial thing I was going to say is that the World Small Animal Veterinary Association funded at Penn a database of testing. So what I think, if a veterinarian is interested in it, they can go to that site and put in dog bladder stones, and it will say where you can get it tested. And I think references, maybe. It's very superficial.
Yeah.
It does not verify, it does not provide any of the information you would say. But to my knowledge, at the moment, that and OMIA, until this is set up, is kind of.
Dog HealthNet has some information about particular tests and how they should be interpreted. So that's another resource. But again, I tell my students every year about these sites, but whether they remember or know how to find them by the time they graduate or need to look them up, I don't know.
And they're superficial. And dogs, I think, has the most. Cats, much less. Horses, even less.
this ISAC panel will tackle all species, so, or committee.
And the OMIA team I know is working on fleshing out the phenotype data, right? There's little to no phenotype data for some of those things, right? What we really care about is the disease, and if we're gonna search it for, maybe it's important in something else, we need more depth of that phenotype data too.
Just a big shout out to the University of Sydney and OMIA, which is what I call it. Sorry, Ernie, pronounce it differently, but it's an amazing resource. Within literally like 6 hours of publishing a new variant, they contact you and ask you to verify the entry. And, you know, I mean, it's just absolutely phenomenal. It's an amazing resource. I think the same way OMIM used to be. Now I feel guilty because they ask me to give money every time I go on there. But and it has all of the literature. So, you know, it's a phenomenal resource.
Yeah, thank heavens for that. If we did not have that, I know we've been going for a while and we do not need to stop, but I just want to check in and let you know we don't feel compelled to stay. And there may be basketball games coming up, those things, I have no idea. So other questions? Yes.
I'll end on a positive one, at least for me. What are you most looking forward to, you know, the next step of your projects or the coming years? Like, what is getting you fired up so that in the immortal words of, you know, you guys don't look so downtrodden?
I'll start since I'm sitting right here. I love the power of being able to strategically use these mutations. I still dream that somebody can come in with a puppy or kitten, we can genotype them, look at their risk and make a plan that may impact the development, maybe it delays it, maybe it decreases the severity of it, that having that knowledge before they have disease can alter the outcome. And that's, I think we're really close to that. So I'm excited about that.
Well, I hope to keep clinical cytogenetics alive with the next generation of students and postdocs and combine it more with genomics.
I'm with Kate, right? I want to see us translate these things to the clinics and not just for the Mendelian diseases. I think for the complex polygenic diseases too, and actually have a little consulting business on the side where I do exactly what Kate's talking about for some dog and horse people. And people are excited for it and want it. And I think there's so much that we can do and how we can really transform lives. I think to Leif's point too, we don't have to know the exact mutation to look at risk. Sometimes it's helpful for biology, but there's so much we can do. And I think I got a Doberman puppy and I messaged Kate and said, what was the supplement that you looked at with PDK4? Because I've got a puppy and I put it on it, and you know, those things are exciting.
Thank you.
Both, thank you. So I'm still new in my career, so I haven't gotten downtrodden just yet, but I know. But so I guess mostly what I'm excited about, you know, in addition to developing polygenic risk scores that can allow, you know, information for owners to know if they should, you know, watch out for disease. I'm really kind of optimistic about machine learning, artificial intelligence, because I know, you know, more genomes don't necessarily help, you know, find the mutation, but the more data that we have and the more training we have on very complex phenotypic data that's hard for us to wrap our minds around, the more we can probably get at the biology, right? You know, yes, risk scores are important, super important.
And they take machine learning.
And they take, right, exactly. So I think there's still a huge potential to get at the very specific interactive biology with enough information.
I look forward to watching people like Brian do these things that he's saying What I'm most enthusiastic about now is that my granddaughters will be here tomorrow.
That's awesome, Gus. I'm also excited about Brian and his future pan-genomes because my obsession for the end of my career is transposable elements and line 1 in particular. And if you've talked to me for 2 minutes, you've probably heard about it.
It's one of the main reasons we're doing it, right?
Thank you. So yeah, I think it's all about the lines in dogs.
From the panel, anybody have any last things you feel like we really didn't say this and we should say this that you would like to?
Just to
Oh,
feed
George.
just to feed the discussion here, maybe before the conclusion. And
No, that's-
I think I'm probably going a bit in the direction of Lev, and I sort of react to your vision about imagining tests which get you into predictive medicine and treatment. So that's sort of really imagining a world that is parallel to what is happening in human genetics, right? So it's sort of not, it's sort of treating, it's still, I think, reactive, and it's still sort of trying to adapt the treatment based on a prediction given the genetic problems that the individual animal has. And while obviously I can understand that that's the vision in human, but it's also inherently, I think, the limitation in human. So for instance, when we talk about polygenic risk score, and Richard tells me I have doubts about the potential of that. I'm interested to talk to him about that. But what I know is that if I look in livestock and not in companion animals, that actually what I think you're trying to achieve is working reasonably well. I mean, you can disagree with me, of course, but I think that the polygenic risk score, that is genomic selection, work remarkably well to change the genetics of a population. So in terms of an individual, it's always, I think that's what the limitations in human, but in terms of moving a population in the direction which you want, it has proven to be remarkably effective. And at least my experience in the population that we work with is that the management of genetic defects is, I mean, it's nothing to, so one of the breeds that I work with, perinatal mortality was over 20%. 10 years ago, and now it's less than in Holstein, just because we manage the genetic effects quite quickly. So I sort of tend to, with all due respect, say, I don't think that's the thing to do. In what you have to do is you have to select. You have to organize the selection, as Lev was saying, to just improve the genetics of the companion animals, no? And I don't know exactly where you want to do that, but
excellent points. Two things. One, I understand your point that it is still reactive in my mind, because they don't have the disease yet. They just have, but anyway, I think you sort of need both, but I think we can't. I think what's a little different about companion animals is they live with many, many, many different people, and they have many different club organizations, the North Carolina one, the Texas one. So it's, I think, a little bit harder, in my opinion, to get them all on board with breeding a certain way. So I think we need to do both. Continue to guide them through education on how to look at risk scores and make intelligent decisions. But I think with these small gene pools, there will probably always be a certain number of animals that have some degree of the genetic mutation. And so shouldn't we, if we can learn to come up with a plan for them, shouldn't we offer that to the ones that are produced?
I don't have any problem if you do that in any way.
Yeah.
You know, it's very hard to predict the future, isn't it? But it seems to me now that from a technical point of view, the easiest thing is, irrespective of all the practicalities,
Yes.
is to sort of select. And I think we could even say we have a moral duty, right? So that's a bit what's happening in I think in Sweden and so on, is sort of to say, if we can, we should breed animals that are healthy for plenty of reasons. And if you have the possibility to breed them, why not do that? I was thinking just...
But there are breeds that, so I mean, we'll take the French Bulldog.
Yeah.
There is no normal French Bulldog. The French Bulldog without all of the mutations that make it look like a French Bulldog is a different breed. It's a Staffy Bull Terrier, right? They have 5, 6 different variants that make their craniofacial defects and their body defects. So there is no room to select or to use selection in that breed without crossbreeding.
Right, I think it's another
Yeah. And so I mean, but there are examples. Guide Dogs for the Blind has selected against hip dysplasia using EBVs. It worked very well. A very large breeding population with lots of litters, 700 litters a year, and it's very successful. But they're one owner and just a few people in control instead of all of the people. But there are some breeds where you can take French Bulldogs and breed them all you want. You will not get, they're homozygous for their variants, so they won't go away.
I would echo what Kate said as a clinician, right? I mean, obviously we want to breed away, and my passion about the Doberman is doing that. I'm passionate about the breed, but I think when you stand in front of an owner that has this one animal, imperfect as they all are, and they want to do everything, that there's also motivation to provide support on that side. I think, they're both important.
It's not mutually exclusive, right?
Right, yeah, exactly.
And just to go, because, we say in French, l'argent est le nerf de la guerre, meaning, you always money for that. And Richard is sort of saying, I think, can I use the model paradigm as a way to raise funds, right? But I just would like to know, in the US, how many purebred dogs, I will say, are being born every year?
750,000 are registered every year right now with AKC, which is the major kennel.
So it sort of be interesting to say, okay, how much money do you need? Because I think this is not so much a scientific program. It's really a breeding program here. And you could sort of say, if I target all the purebred dogs in the US, right? So how could I structure a breeding program like that? Obviously, the breeding organizations are too small individually, most of them. So you sort of say, I need to have an umbrella thing to do that, and participants who want to participate. Then you say, if you want to do that, what's the business proposition here? What's the business plan? So you sort of need to say, what does my annual budget need to be to do that? And I have to spread that over how many purebred dogs every year. So, and do I have a reasonable financial propositions? I'm not sure how much you need, but 750,000 is actually, so every year 750,000. It's good enough.
And now French Bulldogs are #1, so.
Yeah, I mean, I think it's interesting, and what's hard about the financial proposition for dog breeders is that Danika knows, or I know, like, you don't make any money. It's kind of like breeding horses, right? It's not, for the most part, because they are companion animals, there's not a financial win for breeders to provide genetic testing. The ones that want to make a change want to do so because they love the breed. But I can't think of anything, I mean, I don't even want to think about how many tens of thousands of dollars go into the parents of every puppy. I don't know anyone who makes money on it who is doing it well and ethically. I think the people making money are doing it poorly.
Questions, comments? Panel, anything? Anybody feels left unsaid? All right. Thank you all very, very much to the panel. It was outstanding. And the audience engagement, thank you. Really excellent discussion. Thank you for this opportunity. always do a wonderful job. Nobody else does. I know.
So this concludes the symposium, but I think it's appropriate for me to offer a Jim Womack story as we leave. So when Jim was elected into the National Academy, the university had a banquet in his honor. And at that banquet, after he gave his acceptance speech, he was asked to what does he subscribe his success, his academic success. And he said he always tried to associate with people who were smarter than he was. And at this symposium are a lot of those people. And so I want to thank the speakers, the participants, and all of the attendees for making this, I think, an excellent symposium. Thank you very much. Have a safe trip home.